let's start this next section on foodborne illness. Now this is following my sections on transmission of infectious disease where we talked about fecal-oral transmission. So foodborne illness is often one of the targets for preventing transmission of infectious disease. I teach an entire course on food safety, and with foodborne illness, the main thing you're trying to do is stop the transmission of infectious disease. So in food safety, what we do is talk about the different types of transmission in food and some of the different contaminants that can affect food. So there's pathogens from human feces, there's animal feces, there's pathogens in soil, and there's pathogens that can get to you from human skin. For example, MRSA. Pathogens in soil could be from animals and humans. A lot of these spinach outbreaks of salmonella. So salmonella is infecting fresh spinach, fresh cut spinach, or other fresh cut greens, and that may be from animals in the fields. Also with some strawberries and you know, there's mangoes, mangoes of course on a tree, but there's different types of pathogens that will infect different types of produce, and it always depends on how the produce is processed in the factory. So we have this saying with food safety. This is all I'll do for food safety really, but keep it hot or keep it cold or don't keep it. So in other words, temperature danger zone for food safety. You have to have it above 140 degrees Fahrenheit or below 40 degrees Fahrenheit. So in other words, it has to be pretty cool or frozen or pretty hot. And then when you hold a lot of foods, they have to be held for only a minimum amount of time at both of those things. So really what happens is you should have your food held only when it's hot at a lower hot temperature, maybe at 140. Sometimes there's rules that say 440, 140, so then there's 4 hours at 140 or less than 40 for freezing and refrigerating things. And there's different standards for different types of food as far as time and temperature. We're talking about the bacteria growth curve here. So this is important as far as food safety goes. Many bacteria are going to have this first initial lag phase, but then when they're exposed to nutrients over time, after 2 hours, for E. coli for instance, after about 1 and 2 hours, it'll go into this rapid logarithmic growth or exponential phase. So you'll get a lot of doubling of the bacteria in this exponential phase, and then it'll even out after a while. But the exponential phase there is an important consideration for food safety, because if you leave something at room temperature where the bacteria are free to grow and they have exposure to nutrients in the food, they're going to grow very quickly, and food will spoil quickly. Infectious disease is also an important consideration for cancer. So infection can increase cancer risk in a lot of cases. So in other words, a chronic irritation from some kind of infection can cause cell proliferation, and then you can have these types of cells that grow when you can have the pre-cancer cells start growing because of this chronic irritation. There's known infectious causes of cancer, and they account for about 18% of cancers worldwide. The CDC has a pretty good website on this. You can go there and check that out. Liver, cervix, stomach. Also, there's a higher percentage of cancers from infectious disease in lower income countries. So here's another slide about the chronic sequelae of infectious disease. So that just means the way that infectious disease can actually be chronic, which is something you don't think about. You usually think about infectious disease as something that happens immediately. Well, with this infectious disease, you have microbial factors where the environment can combine with different types of human behaviors or microbial virulence factors, and then there's some kind of modified genomics that happens, and then if eventually you get this production of infectious of cancers from exposure to infectious disease. So here's another slide on percentage of cancers by infectious agents in industrialized and then lower income. So we have the developed and developing countries, or we have the disparity here. So in other words, most people that get cancer from infectious disease are going to be in developing countries. So things like this liver fluke, you're going to be much more exposed to that in developing countries than you would in the United States. However, stomach cancer from Heliobacter pylori, there's going to be a lot of that in developing countries, but also a majority of that in the United States. So everyone's exposed to this bacterium, and whether or not you develop ulcers or digestive problems from it, you can get a stomach cancer that way. Here's the we're going to transition now to survival, and I'm not talking about survival of the human from cancer. I'm talking about survival of bacteria or the organism or viruses. And this is another important thing as far as transmission goes and as far as public health microbiology goes, because you have to consider how these organisms actually survive in the environment. Many of them will be vulnerable to a variety of things, but certain bacteria will be resistant to some things. So this is a picture here of seawater, and oftentimes in the seawater, there's certain organisms that can live well and certain ones that can't. Seawater, of course, is very salty, but a lot of organisms can tolerate this degree of salt. Enterococcus is one of those. But then sunlight is something that kills bacteria immediately through a variety of mechanisms, because as you know, there's a lot of different wavelengths of sunlight that comes down from the sky, so from the sun. Also, the temperature can kill bacteria. Oftentimes, bacteria can withstand being frozen. They just kind of go dormant and inactive for a while, and they come back alive as soon as they thaw, but with high temperatures, they get destroyed. Also, adsorption into surfaces, especially with charges associated with those surfaces, they'll sometimes be stuck there forever and then be, therefore, inviable and not be a threat. A lot of different things to consider for survival. Some factors that make them resistant to some types of water treatment. Some cell walls contain a waxy material. Others that will allow the cell wall of the bacteria, for instance, to persist a little bit longer. MRSA, for example, has more of a waxy material on its cell wall. Some of the parasites have a thick-walled protective stage, oocysts. So, for example, Cryptosporidium is a water parasite, and that'll have a thick-walled protective stage. Also, a lot of them will have a small size, they'll clump, and they'll associate well with organic particulate matter. They have a lot of different factors that help them survive a little longer. Some of the most some of the most resistant organisms to different things, so you can have a lot of different processes and things to kill organisms out there. Cryptosporidium is a pretty hardy parasite organism. It's a protozoan, and it is resistant to chlorine and also ozone and iodine and some other halogen-based disinfections. Ultraviolet light, some of these viruses, adenovirus and rotavirus, are resistant to that. While we have ultrafiltration as well, and you have enterovirus and norovirus that are resistant to that, and that's because of the size. Those very small viruses. Mixed media filtration in general, most filtrations, you're going to have viruses that slip through because they're so small. Wetlands and ponds, a natural wetland system or a constructed wetland system, those are viruses. Those are very, very fine filters, and so viruses will be able to slip through those. And thermal processes, there's also some viruses that can slip through that too, depending if it's a low temperature sterilization. So say you're pasteurizing something, or you're disinfecting a type of bacteria that you might only have to heat up to 160 degrees or something. Then the water may not boil, but you're supposedly getting the kill temperature. Well, for some viruses, they can withstand that. Again, here's survival times in water, wastewater, and soil on crops and on crops. Um, so these are um, different organisms. If you look at the bottom there, you have Ascaris lumbricoides. That is a roundworm. That is actually a helminth worm, and it has a really, really tough egg. So it produces a bunch of eggs when it's inside the human host, and those come out in the feces. And then, so if somebody is passing their feces in a field where they're growing crops, those eggs can stay around and really contaminate things. When I was in Ethiopia, I did my Master's of Public Health in Ethiopia, actually here in Loma Linda, but did my field practicum there. And I collected 1,000 stool samples and processed them, and roughly 60% of everybody had Ascaris in them, living in them. And then there was also Ascaris was the number one. There's also a lot of hookworm and and Giardia that we found. So these are some of the survival times of different pathogens.